If people could take their seats. This is one of the really fun parts, the most enjoyable parts of Chaos, which is the short talks by the 5 people who won poster awards. And I want to remind them that because we're having 5 talks in a short period of time, that they have to really keep to schedule. Each of the speakers will have 12 minutes in which they can tell their story with 6 minutes discussion. Okay, so we'll get started. Our first speaker is Avital Chachami from the Weizmann, who is going to talk to us about whatever shows up on his screen in a moment or two.
Hi, my name is Avital. I want to thank the organizers for this wonderful opportunity to tell you about how the blind see. This is a work done in collaboration with Meital Wilf, Boris Rossing, and Fruman Haran, Marlene Berman, and Rafael Malach. So throughout my talk, I'll be talking about spontaneous brain activity. And that is basically what happens in our brains when we do nothing in particular, when we simply rest. And we know quite a bit about this activity. We know it's ultra slow. In fact, what you see here is 3 times faster than what goes on in our brains when we rest. We know it appears across the entire brain, and it also has a proposed role in reflecting the statistics of brain activity as evoked by Our Lady behavior, which is a passive role. But given that this is brain activity, it may have an active role, and it has been proposed that it could underlie conscious cognitive behavior. So these authors suggested that neural networks have some theoretical ignition threshold. So when a stimulus appears, it could evoke high amplitude fluctuations that cross the threshold, and then the network enters into an active mode. Then stimulus-related behavior follows. But they also claimed that the stimulus is actually not necessary. So even when there is no stimulus, the very slow spontaneous fluctuations could occasionally cross the threshold, and the network would be active again. So this very slow accumulation of activity. Should I just take a step back? Okay, so this very, no, stop. Should I continue? Yeah, is this okay? Cool. So there is a very slow accumulation of activity that may be taken as a marker for the contribution of spontaneous activity if we see it before the onset of behavior. And given that this behavior is not driven by a stimulus, we could refer to it as spontaneous behavior. Now, how can we empirically test whether spontaneous neural activity underlies spontaneous behavior? This is a very hard question, because on the one hand, you can't ask participants to be spontaneous because by the mere instruction, you're losing all spontaneity. On the other hand, you can't have a task, because if you have a task, your brain doesn't act in a spontaneous way, but it's task-evoked activity that you see. So to address this question, we decided to study blind individuals, because in these individuals, there is no external visual evoked activity in their visual cortex. And we chose those individuals that have visual hallucinations, and these can be as vivid as typical vision. So the reason I refer to these as spontaneous is because neither the appearance nor the content of the hallucinations is controlled by these individuals. This condition is termed the Charles-Bonnet syndrome, CBS, and it's defined by visual impairment, visual hallucinations, no psychiatric disorder, and insight. And insight means that these individuals know that they are blind. They know it doesn't make sense that they can see, and they're therefore afraid to be tagged as insane. And usually they don't tell anyone about their condition, including their family members. Obvious, it's very hard to recruit them. So we found 5. I want to describe their hallucinations to you now. The first one hallucinates face. It's a face as real as you looking at me now. It's always the same face. The second one hallucinates different patterns and faces that can be either real or ghost-like. The third one hallucinates still images from different categories. Now, bear in mind that these individuals can report their hallucinations because they're late blind. They previously saw. So when they report, they rely on their experiences as sighted individuals. We also recruited 2 individuals who cannot report their hallucinations because they're just too fast. The first one hallucinates very fast flashes of light, and the other one hallucinates something like a kaleidoscope. We also recruited 11 blind controls who have never hallucinated and sighted controls. So what are the neural correlates of hallucinations in CBS? Actually, only 2 studies have addressed this, and both have ascribed hallucinations to isolated brain activity in the vicinity of the fusiform gyrus. But if you think about it intuitively, I mean, if hallucinations are just as vivid as typical vision, you kind of feel like it should be more areas that are active. And even if this is true, if you have one region that is active, by the mere fact that it's functionally and anatomically connected to all other areas in the visual system, you should see some spread of activation across the visual cortex. So unlike previous report, we hypothesized that in the spatial domain, visual hallucinations like veridical vision would activate the entire visual hierarchy. As for the contribution of spontaneous brain activity, a recent study done on CBS participants who are only partially blind has actually ascribed hallucinations not to spontaneous brain activity, but to external visual stimulation. On the other hand, this study that I cited here actually showed a slow buildup of activity prior to the onset of hallucinations. And this, as I said, could be a marker for spontaneous brain activity. So here we hypothesized that in the temporal domain, hallucinations would differ from the vertical vision in the spontaneous buildup of activity prior to the perceptual event, and this would not appear only in one area, but across the entire visual system. So we asked CBS participants to verbally report their hallucinations while being scanned in the fMRI scanner. And we constructed this log of their experiences, and we asked them after the scan to provide as much detail as they can about these hallucinations. And based on this detailed report, we created simulations of their hallucinations, which we presented as movies to sighted controls who were also asked to report verbally report what they see. Now, bear in mind, there is no possible way to accurately represent hallucinations. We can only be as accurate as choosing the right visual categories and be as accurate in the timing of the onset of hallucinations as reported by participants. This is an example of the hallucination stream of the first participant who hallucinates a face. It's really quite boring. This is an example of the second participant hallucinating different patterns and faces. And this is the third example for the participant who hallucinates different visual objects, categories, people, many different things. So CBS participants, when they hallucinate, have spontaneous vision, whereas sighted controls, when they see these hallucinations, have non-spontaneous or cued vision, because we present the stimuli, we decide when to display the stimuli. But this is not the only difference between these conditions, because in CBS, the vision is internally generated, and in controls, it's externally generated. So to account for this difference, we included another condition, which is visual imagery. And here we have cued vision, because we tell participants when to imagine, but it's also internally generated. So the only condition when we have spontaneous vision is in CBS during hallucinations. So across these conditions, we expect to find activity across the entire visual cortex. But unlike these non-spontaneous vision conditions, where we expect to find the percept and then a rise in signal, here in CBS during hallucinations, we expect to find the rise in signal and only then the onset of hallucinations. So this is our spatial hypothesis. This is an unthresholded map. I'm sorry it's flat. Basically what you see here is anterior, posterior, dorsal stream, and ventral stream. And what you see here in contours is areas that are active during hallucinations in CBS, and they basically span the entire visual system. We can look at simulated hallucinations in sighted controls, and these activate a different part of the visual system, which is more foveal, and this is because of experimental constraints. Our stimuli had to be very small. Nevertheless, we can superimpose these contours of hallucination onto the sighted controls map, and we see some overlap here, which we can quantify by pattern similarity across the entire the entire posterior part of the brain. And when we do that, we find significant similarities between hallucination-related activity and visual activity in typical vision. We can also look at imagery. So for the CBS participants and blind controls, you can see that imagery activates the visual system. And when we superimpose the contours of hallucination over these maps, you can see a very nice overlap. It reaches significance for the blind controls, although it is of the same magnitude in the CBS participant, but it may be that we're just underpowered to find significance there. So it seems that across all these conditions, hallucinations, like all other types of vision, activate the entire visual hierarchy. As for the temporal hypothesis, here I remind you, we expect to find activation in negative time points, that is, prior to the onset of percepts, just a slow buildup of activity. We tested this in 3 ROIs, an early visual cortex defined as V1 to V4, the FFA, and the lip area, which is a control area because it's engaged in a task because it's a verbal task, but it shouldn't show this accumulation. And indeed, we find that in early visual cortex, CBS participants show an early rise of activity prior to the onset of hallucinations, which we don't find in typical individuals who just have vertical vision. There is no group difference in the imagery between groups. For the FFA, we see again a similar effect, although reduced in its magnitude for the CBS participants, and no group difference in the imagery condition. And for the lip ROI, we see no group differences across either of the conditions. So it could be that hallucinations indeed differ from other types of vision in the buildup of spontaneous activity prior to the perceptual event. Finally, is this signal accumulation uniform across the visual system? To address this, we ranked visual areas according to their serial position across the visual hierarchy. And we measured the time where they start accumulating their activity relative to the onset of hallucinations. We find that this accumulation of activity is apparent across the visual system, but it's more pronounced in early visual areas than it is in higher visual areas. This is not the case. We don't find this relationship for sighted controls during simulated hallucinations or in CBS participants during imagery. So it seems that there is an anticipatory spontaneous activity that decays along the visual hierarchy during hallucinations. So I want to add in some outrageous speculations, if I may. Here we try to understand whether spontaneous neural activity underlies spontaneous behavior. We studied this in blind individuals who experience visual hallucinations. Based on our results, we can hypothesize, and this is only one possibility, that perhaps in these individuals, due to deprivation, the ignition threshold is reduced, and therefore the visual system is susceptible to noise. And therefore, random spontaneous fluctuations, even if they're not of high amplitude, could cross the threshold, spread through the visual system, and bring about hallucination. This may not only be the case for blind individuals, but also for sighted, because it has been reported that after deprivation, actually short deprivation, visual individuals can actually experience hallucinations. And if we don't care about deprivation for a minute, you can think about a situation where spontaneous activity may also accumulate and ignite behaviors such as dreams. Or in different networks in the brain, perhaps if we have accumulation of activity in the motor system, then we would have spontaneous motor activity. But this is, of course, mere speculation that needs more work in different projects. So I want to thank all my collaborators and funding agencies that supported this work, and thank you for listening.
Thank you.
Epileptiform discharges in a part of the brain that spread and don't spread to the whole brain. And the phenomenology that's associated with that in the part of patients can sometimes be visual hallucinations when the simple partial seizures are in the visual system. I'm just wondering if it could be the case that these individuals have a very mild form of epilepsy, and has anybody ever looked to see if actually that is what's going on?
From the clinical literature, I think there is no comorbidity. As for our sample, these individuals are completely healthy. They're actually most studies done in CBS are on very old individuals, like in their 80s. Here we had the opportunity to actually recruit young individuals. They're like in their 40s, and they have no neurological or psychological, I don't know, but psychiatric disorder that we know of that they have reported.
Well, just to follow up, I mean, maybe that is exactly it. I mean, you know, that's the question is, and you wouldn't know unless you did the right kind of EEG experiment to test, right? I mean,
sure,
and sometimes patients can have exactly just those kinds of symptoms and everything else is normal.
That's very interesting. Yeah. That would maybe explain why it doesn't happen to all blind individuals who lose their vision. So currently we don't know why it is only, by the way, the prevalence of this is between 0.4% and 14%. It may be much more because they don't tell anyone about it. But this could be the reason why it happens only to some individuals and not to others. Yeah, I don't know.
Any other questions? I have one question, which is, I take it these are people who lost sight after they've had sight for a considerable period of time. Sorry, I already asked her. Okay. Sorry. All right, so moving on. Thank you very much.
So thanks, members of the committee as well. It's a great honor to be here doing this. There's also a bit of irony here. My background is not in motor control, but I did go to grad school at Western, where Mel works, of course, and I participated in most of his experiments. So I'm not an authority on motor control per se, but I am an authority on how awful these experiments are to be participants in. At times they're truly. Yeah, it's, but it is, you know, it's fun to be here, you know, talking about reaching in front of Mel, since I overlapped with him for a long time at Western, and I didn't do any of this work there, so. So I guess, having said that, I mean, the punch, I guess what I'm going to give away is that in chaos, actions, so I'm going to inject actions into the meeting so far. And specifically building on, like Mel suggested, building on some of the work that Morgan talked about in the previous, in the previous session. And specifically, I'm going to show you how you can use action to reveal hidden cognitive states that relate to processing of object concepts. So much of our adaptive behavior really reflects the contents of semantic memory. So we have this idea of object concepts. This is our knowledge about objects, and it's what it means that it's the content that means we don't have to rediscover the properties of an object with each subsequent encounter. So if you were to sit at this desk, you would have no problem picking up the pencil and writing a note. You have knowledge about pencils, you have knowledge about paper. This comes to mind effortlessly. But we live in a more complex world. We have desks with many objects on them. And what this means is that on occasion, we have to resolve some competition between these different objects to achieve some goal-oriented behavior in an efficient manner. So for example, here you might need to discriminate between the phone and the pencil and the keyboard, and for some reason this other phone this person has, if you wanted to take a note, right? These are all things that you could use to take notes, so you need to resolve this ambiguity. And there's a lot of really interesting research, some of it from Mel's lab in humans, as well as from electrophysiological recordings in non-human primates, showing that you can get some insight into this decision-making process by looking at visually guided action. And it's thought that your hand, as you move through space, can reveal this decision-making, and it can reveal the resolution of the competition between these different objects. So this is the way that these experiments typically look. I'm probably generalizing, well, I am generalizing quite a bit. This is how the one I'm going to show you looks. The basic idea is that you set up, you could use this, so the way that I've done this is using a touchscreen display. You could also use this using mouse tracking, and you could do 3D motion capture. But the basic idea is that the participant starts at one place, I'll call it the home location, and then they're going to be reaching out and touching one of two targets. There could be more than two, but for simplicity, there's two targets. And the critical point is that one of these, during the initial stage, they don't know if they're going to the right or to the left. So there's ambiguity about what the target is going to be, and that's important because that neurophysiological work has shown that when there's target ambiguity, there's multiple competing action plans in dorsal premotor areas. And as a consequence, the initial trajectory of the reach is sort of at the midline. It's average between the spatial location of the two potential targets. So the idea, though, is that one of the locations is cued as being the target and the other is now the distractor. So this is what two reach trajectories might look like in a given task. So we have an orange path and a blue path, and let's say that both of them have the same target, but they differ in terms of what the distractor is here. So we can look at this path through, in this case, two-dimensional space to interrogate the nature of that decision-making process. So we can see here, for example, that early on these paths are almost identical, but at some point they deviate from one another. So the orange path has this earlier deflection toward the target and the blue path is later. And then we can ask then, how does that relate to this relationship between the distractor and the target? And what I'm going to be talking about is looking at how the visual and conceptual similarities between different targets and distractors can shape this trajectory and what that might tell us about the decision-making process. So just to be clear, when I say visual properties, I'm talking about visual semantics. So the content of semantic memory that is related to the way things look. And when I'm talking about conceptual properties, I'm talking about more abstract information that's associative in nature or things like function. So the way that you can look at these data, there are many ways that you can look at these data. One of them is somewhat crude, is just to look at area under the curve and just compare that for the two different trajectories. So you can see here there's less area under the curve for the orange case than there is the blue case. And that tells us something about the nature of that decision-making process, namely that this one is resolved, the competition is resolved earlier. Another thing we can do is look at where, in comparison to this midline, do we see the first evidence of statistically reliable deviation from the midline. So these are the two metrics I'm going to talk about today. So the critical question, though, that I'm interested in is about this relationship between the distractor and the target. So to get at this issue, we set up a study that goes as follows. There's basically 3 stages to each trial. Each participant completes 400 or so trials. The first stage, they're just putting their finger on the touch screen, holding it there for about 5 seconds, and then above 2 potential target locations, there'll be 2 words appearing. These are referenced for object concepts. Here it's phone and pencil, for example. Once those words appear, the participant has anywhere from 2.5 to 2 3/4 seconds. We jitter it so they can't predict what they're going to do next or when they're going to do what they do next. And during that time, they have to read both of those words aloud, and we counterbalance which side you're reading first. So the idea there is just we want to make sure that they're engaged with the stimuli. And then, after, again, this short delay, the competition is resolved, so to speak. So this color coding tells you where the target is and where the distractor is. So in this case, this participant is always reaching to purple. They're never reaching to yellow. So now they know that this is the target, this is the distractor, and what we ask them to do is make a really rapid reach toward the target. So the critical point here is that they only have 500 milliseconds after the cue. Well, they can go beyond that, and in fact, sometimes they do, and I think that's interesting, and I'll show you why. But we encourage them to try to respond as quickly as they can. And when they do respond in less than 500 milliseconds, the mean time from holding that point to initiating movement after the cue is about 380 milliseconds. Once they've initiated that movement, they then have 700 milliseconds to touch the target. So again, this is all happening very quickly. They see the cue, they're reaching out almost instantly and then touching the target, and it's all happening rather quickly. So to get at the critical question here, and Morgan talked about this at great length in the last session, what we wanted to do here was parameterize how the degree of similarity between the different kinds of distractors. So we want to have a highly similar visual distractor, a highly similar conceptual distractor, and so on and so forth. So we collected a fair bit of pairwise ratings from a different group of participants than Morgan talked about. But to give you a sense of what that looks like, so let's say that the target on a given trial is hairdryer. A highly similar visual distractor would be gun, and a highly similar conceptual distractor would be comb. So in this case, the link here is that those are both used with hair to style hair. They're found in the same sort of locations. The visual distractor is just based on the fact that they have a similar form. And then in a second bin of items, we identified another range that we call medium similarity. So here the medium similarity visual distractor is megaphone. For those of you who don't know what that is, it's like the big speaker that you speak into that sort of cones out. Not a great description. And then the medium similarity for conceptual distractor is scissors. Again, the relationship is hair and hairstyling. And then we have a low similarity bin where these things have no basically no conceptual similarity on the visual distractor side of things, and likewise no visual similarity or conceptual similarity in the conceptual distractor. So the point here, though, is like we did in the paper Morgan's described, we have made sure that the visual distractors are not also conceptually related in that more abstract sense. So any effects we see, we think are not necessarily, we don't think we can attribute them to the other type of similarity, I suppose. Okay, so these are the data. I'm going to show you first for instances where they're resolving competition from a visually related object, and then for the instances where there's conceptually related objects. And I'm just going to plot the area under the curve here. So for low, medium, and high degrees of similarity. And what we find is that the visual similarity of the object among the object or between the object concepts doesn't seem to influence the reaching trajectory. But we see a very different pattern when we look at the conceptual similarity. So here we see this nice linear trend where as things become more conceptually similar, the bias toward the distractor is increasing. So the time it takes to deviate from that midline is increasing. And I can show you what that looks like. These are the actual trajectories, mean trajectories across individuals. And I'm plotting in cases where there's a visual distractor going left and a conceptual distractor going right. Of course, in the actual task, they were going both ways. And what you can see is that for the visual distractor, they're basically the same across the entire range of the trajectory. And in fact, they deviate from that midline point all around just over halfway through the reach. And we can see what I was showing you with area under the curve in the conceptual distractors, where the low similarity instances, you see these deviations early on in the reach, about halfway. Medium similarity a little bit later, and then when there's a high similarity distractor, you see this they're really sort of persevering down the center until they can resolve that high degree of competition. Okay, so I talked about this as being a rapid, visually guided reaching task, but there were also cases where it wasn't so rapid. And what I specifically mean is during this queue period. So we encouraged them to move as quickly as they could, but they didn't always do that. And it's hard to say exactly why, but there were certainly a subset of trials, in fact about 37%, where the movement took significantly longer than that initial threshold that we had of 500 milliseconds. And the mean was 722 milliseconds. So there's a difference of about 400 milliseconds between these two bins of trials, between when they're moving quickly and when they're moving more slowly, to initiate the movement anyway. So we thought, well, maybe we can exploit this and see what's going on. Here, the idea is that they've probably resolved the competition between the distractor and the target before they've even initiated their reach. So again, this is area under the curve, and we see that in the case of visual competition, so these darker bars are what I showed you earlier. The lighter bars are the slower movements that I just described, and we see that there is a main effect. So if you take more time to initiate your movement, you're going toward the target in a straighter line. But again, there's no influence of the degree of similarity on that reach trajectory. And this is what I think is really cool about these data. We see this beautiful interaction in terms of conceptual distractors. So whereas when they're moving really quickly, we saw this linear trend upwards. So as similarity increased, so too did the time that was required to resolve the interference. But when they take more time and maybe are more deliberate with their actions, we see that a nice linear trend that's trending down, whereby as things become more conceptually similar, people are taking a straighter path to the target. It's almost as if they're recognizing that this is a high-stakes trial, and it's really important that I avoid this really highly related distractor. So I'm going to be really careful and go straight there. Whereas when there's less on the line, maybe they're playing a little more fast and loose with their reaching. So these are just showing you, again, the reach trajectories. And you can see here that the high similarity condition is deviating from the midpoint about 26% of the way through the reach, which before, in the other, not condition, but when they were moving more quickly, that was up here at 77%. So there's this really, really dramatic flip that's happening. And I think it's telling us something about how resolving competition amongst object concepts in a reaching task is telling us something about the importance of that sort of cognition on efficient motor control. So the take-home message, a friend and colleague and collaborator on this project, I've taken this directly from him, Craig Chapman, who used to work with Mel. He likes to say that moving is thinking, and I think that that's true. Here I'm showing you that reaching can reveal hidden cognitive states, and specifically cognitive states related to this decision-making process between object concepts. Second, the time required to resolve competition between object scales with their conceptual similarities. We didn't see anything in terms of visual similarities. That story might be different if we use pictorial stimuli. And then lastly, resolving competition optimizes reach efficiencies. Thank you.
So that was very nice. I was wondering about the sort of asymmetry between the visual similarity and what you call conceptual similarity. And I wondered if you'd like to speculate about that and whether it might be to do with the fact that the visual properties afford the motoric interaction, and whether there may be a sort of interference between that and so I'm trying to think. I'm thinking of the kind of work that Eiling's in fact done, showing that your retrieval of highly visually experienced concepts is, no, not visually, motorically experienced things is interfered with if you have to do a motoric movement at the same time. So I'm just wondering whether you think, what you think is the cause of the lack of low, medium, high similarity in the visual, for the visually similar things.
Yeah, it's a good question. We did choose objects that all are graspable. They're all objects that you would use. They have a function. I think what this is telling us is that there's a certain primacy in terms of the functional properties, and those are more abstract in nature. There's certainly affordances. In most cases, they are closely linked to the functional properties, but not always. So I don't quite know. My suspicion, though, is that if we used images, the story might be a little bit different, and we might even see an interaction. So I don't want to claim that the visual information doesn't matter. It clearly does matter, only that in this task, where the decision-making is about object concepts presented with words, it doesn't seem to play that big a role. But I wouldn't say that it doesn't play a role at all. It's hard to speculate, really, on what that difference is. Yeah.
Okay, so I think I missed something in the description of the task, and it's partly why I don't understand, but how were the targets on each trial determined? Because you couldn't have it in the same location each time, because then people would simply reach straight there. So they have to do some kind of discrimination or evaluation of the words to figure out. So what is the rule for determining a target on a given trial?
It's the color coding. So it's the color coding here. So initially the potential targets,
I see. So you actually cue with the color on, okay, got it.
Yep.
All right, very good.
So the processing of the word, I mean, we have them read it, but it's largely incidental to the actual task.
Okay.
Yeah.
No, it's fine, it's fine. It was very simple.
Just following on Jerry's question, I just wanted to suggest two hypotheses for why you might not see the visual effects and just get your very quick reaction to them. Sure. So one is similar to what he was saying, which is given that they don't actually have to grasp the object, the reaching behavior is kind of in conflict with any stored information they have about the shape because they're doing a poking motion, right?
Yeah.
So it could be that you actually need to suppress that information. The second possibility is related to work that I'd shown years ago with semantic priming, and then along with Eiling Yi, we showed with eye movement data, which is that the visual information comes online earlier and then goes away. So you have this fairly long period. So it might be that you've missed the activation of the visual information. Yeah. Either of those seem more likely to you, or
I think both are both. I could see it going either way. Honestly, one interesting thing that we didn't do here. There's such thing as a go before you know task. And that's when you initiate the movement, and only after you're in flight do you get the cue to what the target is. So I think maybe if that was done, maybe it would reveal if it's the visual is earlier. That might be a more sensitive way to look at that. I'm not sure though. Yeah.
Hello, everyone. Today I'm going to be telling you about some research looking at the role of experience and how experience can influence cognition and brain function. And the way that our lab group, the Bedney Lab, gains traction on this question is by comparing the brains and behaviors of people who have radically different life experiences. So in our case, people who were born blind versus sighted people. So by comparing these two groups, for instance, you can look at the cognitive role of a given brain region and how this can be impacted by cognition and experience. So in our case, we're looking at visual cortex. And there's been a host of neuroimaging studies that have looked at how visual cortex what functions it performs in blind people. And what's been found is that in blindness, the area of the brain that would otherwise process visual input, it starts to respond to non-visual input. And this has been shown across a variety of tasks throughout the cognitive hierarchy, so both for these more low-level perceptual tasks, like localizing sounds and perceiving auditory motion, as well for tasks that require manipulating lexical and numerical information, like these language and math tasks. So there's a whole bunch of results in this field. But one thing is that a lot of these different studies, they use more conventional methods, and they use very targeted tasks to look for specific effects. And because of this, the state of the literature is somewhat piecemeal. So there's a lot of open questions that we can ask about this functional profile. So I'm just going to pose a couple that I'll try to address today. So for starters, this kind of effect that you see in visual cortex of blind people, one question we have is how consistent this putative reorganization is across different people. So does the same effect get mapped to the same area across different blind people? Another question that we're trying to figure out is to what extent the information that gets reorganized into visual cortex, to what extent is it spatially distributed? So we know in the sighted brain, for example, that there's this retinotopic map of information in V1 such that adjacent regions of the visual cortex are encoding adjacent regions in the visual field. And we know that this layout of information across space, it's systematic across different individuals. So an open question is what's happening in the blind brain? So in addition to these questions, we also just want to ask fundamentally, what type of information is being encoded in this region? So if there is some kind of systematic layout that's distributed across space in different people, what kind of information is being encoded in these spatial patterns? So to address these questions, instead of using a more typical conventional design where we might play different beeping sounds for participants or have them listen to short spoken sentences, we measured brain activity while individuals listened to auditory narratives. So we have naturalistic clips from movies. And instead of modeling a contrast to specific task conditions, we're going to directly compare the brains across different individuals while they listen to this stimuli. So I just wanted to give you a sense of what this sounded like for our participants. So I have a 20-second clip that's part of a 6-minute-long excerpt that our subjects listen to in the scanner. So this is from the movie Blowout, featuring John Travolta. It's from the 70s. So just imagine that you're in the scanner and this is what you hear.
Just 10 minutes.
Okay, come on, let me take this.
Okay, thanks.
How you feeling?
Pretty good.
Stopping at Trenton,
nice.
Newark, New York, Rye, Stamford, New Haven, New London, Providence, and Boston. Stairway 3, track 4.
Look, I just didn't want you to go.
Why?
Because I like you and I just met you and you're going off on me.
Okay, so stimuli like these have been used extensively in the sighted brain, and it's been found that if you use these intersubject correlation methods that we heard about yesterday, you can actually look at the time course in each individual voxel and see across different individuals, how does that time course synchronize across people in response to the stimuli. So we're going to take advantage of this method today in order to understand what's happening across different people. And so for our particular stimuli, we chose 3 different movies. So again, these were movies that were originally shown like audio-visual format, but we're just going to play the sound clips for our subjects in the scanner. So we have 3 different movies, and these sound clips are 6 minutes each. And so they hear them one at a time in separate scanning runs. And in addition to these movie stimuli, subjects also listen to a supposedly funny stand-up clip. I think that's debatable. But it's this story about a guy and like what college was like for him, and he just kind of goes on and tells some jokes. But in addition to this stand-up comedy routine, we also have 2 different versions of it as well, where the high-level narrative content has been degraded. So it's the same stimulus in some ways, but it can serve as a control. So in one of the cases, PyMan, we've retained some of the linguistic information such that the sentences are all presented as they would be in the story, but we've scrambled the order of the sentences. So now we still have the linguistic information from the stimuli, but the narrative content has been degraded. And then in this case, we just play PyMan backwards. So now we have the acoustic information being preserved, but there's no kind of narrative content. So across these different stimuli, we have a range of cognitive complexity. So we can test which different stimuli elicit shared responses across our subjects and visual cortices. So this is what the protocol looked like for our subjects. So in our case, unlike in Avital's case, all these people were born blind, so they've never had vision. Their loss of vision is due to pathology that's in the eye or the optic nerve. So they don't have any brain damage, but they have at most minimal light perception, and they don't have any measurable visual acuity. And then we also have sighted people matched on age and education. So everyone goes in the scanner, we make sure that they aren't familiar with the clips already, and then after they get out, we make sure that they could actually understand what they listened to by having them answer some questions about the plot. So in an initial analysis of this data set, we found a really exciting effect, which is that in the visual cortices of blind people, the different individual voxels have a synchronized time course of response across different people. But this is only in response to the stimuli that contain meaningful content. So it's only for the movie stimuli and the stand-up comedy routine, not if it's just backward noise. So this result suggests that there is some kind of shared signal across different blind people in this region. However, from this result, we don't necessarily know whether this information is distributed spatially. So it's possible that in blindness, visual cortex acts as a monolith, such that all of the voxels are responding in a similar way across time. But an alternative possibility is that there's some kind of information distributed across space, and that perhaps different segments of our narratives can elicit a distinct spatial pattern, and that spatial pattern might match across different individuals. So that's what we're going to test for in this talk today. Okay, so just to give you a demonstration of how this is done, let's return to an example that we heard from Janice yesterday. So just to remind you, in Janice Chen's talk, she talked about this beautiful data set that she collected where sighted individuals watched this really entertaining episode of Sherlock. And what they can do with this data is look in a given subset of voxels. So for example, in this posterior medial cortex, ROI, they can look at the spatial pattern across voxels and say if you take the whole subject group, all 17 people, and you break it randomly into half. So this is what the patterns look like for these different scenes in one group, one random half of those subjects. And we can see that if you do the same thing for the other random half of subjects, there's a lot of similarity in the patterns across the 2 groups. And the scenes elicit distinctive patterns from one another. So again, this is just during the movie viewing data. Janice talked extensively about what happens during recall. But in my case, subjects are only encoding the stimuli anyway. So this is most analogous to what I'm doing. So then we can see that if you look in this given ROI, you can actually plot the similarity between individual events. And you can see, indicated by the height of these bars, that there's a lot of shared spatial patterns across these 2 random subject groups. And then again, if you do the proper statistical thresholding, you can make a map, a searchlight map of information to see where in the brain you get these shared spatial patterns across different people. And of course, in this case, it makes sense that they see a lot of shared spatial patterns in visual cortex because their subjects are sighted. But we wouldn't necessarily expect that for our subjects. So let's walk through a quick demo of how this has been working out in the data that we have, where it's blind and sighted people listening to the movie clips. So in this case, let's just look, for example, in a given ROI. So I just sampled V1 from an anatomical atlas, and I took one of my movie stimuli, the taken clip, and I took the entire 6 minutes and I split it up into events, or what I'm calling events, but really they're these 10-second long segments of activity. And so what we can do is for half of our blind subjects, like half of them, and then we have the other half, we can directly compare the patterns of activity across these two subject groups. So we'll do this for both the blind and the sighted. So we have blind to blind and sighted to sighted. And what you can see when we do this is that there's a lot of consistent activity in the blind visual cortex across these two randomly selected subject halves. But you don't see that kind of consistent pattern for the sighted subjects. So we're seeing some kind of shared information in this spatially distributed pattern in our blind subjects in this brain area, which is pretty exciting. You see effects like this in those high-level narrative content areas in the sighted brain, but we're seeing it in this area that would otherwise process vision. But when we did our analyses, we actually did it in a more formal way such that we could sample across the whole brain, not just this one ROI, and we also looked at baseline comparisons for thresholding purposes. So let me just walk you through how I did that before I show you what those searchlight maps look like. So in this case, again, we have our stimuli that we segment into these 10-second long events, and what we want to look at is the characteristic activity pattern that's elicited by each of these individual events in the movie. And we want to look at this for one subject versus the average pattern in the rest of their group. So this analysis is done separately in the sighted subjects and the blind subjects. And we also want to look at how similar the patterns are when the events mismatch as a baseline similarity measure. And so here's what it looks like in an example searchlight. So what I'm posting here is a similarity matrix where we have all of one subject's event patterns and then the rest of their group, their average pattern. So we're interested in this diagonal to see how similar do the patterns look when you're looking at the same event or when you're hearing the same event versus a different event. And so we're doing this iteratively throughout the brain in a searchlight procedure. And we're looking at permuted shuffled pairings to see if it's above baseline. So I have a bunch of searchlight maps to show you, but just to help get you oriented, we're going to look at the data in order of stimulus complexity. So we'll start with that backward sound case where it's noise and it doesn't have any meaningful information. And then here at the bottom, we have an average searchlight map. It's a composite average across all three of those movies, but I also have the individual maps if you want to see those later. So what we can see just for the sighted people is that you only see those shared spatial patterns in auditory cortex. And this is to be expected because they're just sighted people and they're just hearing noise. But as you look through the increasingly complex stimuli for this subject group, you can see that you start to see shared spatial patterns in some of those high-level narrative default mode network areas that Janice told us about yesterday. And that, of course, if you look at the ventral view, you don't really see any shared pattern similarity in visual cortex, which, again, is what we would expect for this control group. But it gets more interesting when we look at the blind data, of course, because we can see that while there is that consistent response in some of those auditory cortex regions, as well as some of those default mode network regions for these more complex stimuli, we can also see on the ventral view that we do have shared spatial patterns, but only for these more complex stimuli. So if this information was organized in an idiosyncratic way across different blind people, you wouldn't be able to find this shared spatial layout. So it's actually a pretty exciting effect for us. And when you look at the difference maps between the two groups, it's only these complex stimuli that show this kind of shared spatial pattern in the blind and not the sighted. So just to wrap up, if we return to our original research questions, we can see that it does look like information is spatially distributed across visual cortex and that reorganization is systematic across different people. And it also appears that it's these more cognitively complex stimuli in particular that can elicit those kind of shared spatial patterns. But just to close, I want to say that we want to delve further in to understand what kind of information is encoded in these shared patterns. So what are the features of our stimuli that might be driving these effects? So to test this, we're using an encoding model approach where we try to tag each one of our different movies according to a bunch of different features that we think might be contributing to the effects that we observe. And then we can build a model by learning the mapping between a set of training data and what the features are at each point in the video, or in the movie clip, and we can try to understand what the pattern might look like. So we actually generate patterns of activity based on the features in our model, and we can see how well those correspond to what we actually observe. And from this, hopefully we might be able to infer what kind of information is encoded in this region. So it's still early, but it looks like maybe some of the top predictors in our model are things like the presence of language and the presence of music. But again, it's hard to know exactly how to interpret those kind of effects, so this is going to take a lot more work. But in closing, thank you all for your attention, and thank you. It's been great to be here.
That was super interesting. It seems to me that in order for there to be a relationship with visual cortex that actually shows systematic spatiotemporal structure like that, there has to be a systematic mapping between the maps in auditory cortex and the maps in visual cortex. And I wonder if you've thought of doing something like, given that we don't know as much about that organization, at least at the level of fMRI and auditory cortex, I wonder if you could go the other direction and take something like Jeff Aguirre's sort of map of V1 that you can use just based on anatomy and FreeSurfer and try to map back to auditory cortex to see if you could see what that map looks like.
Yeah, I haven't thought too much about auditory cortex in particular. In general, when I think about what this region might be doing, I think more of some of these higher level regions, such as precuneus and angular gyrus. But I like your idea of taking this approach of figuring out like what the connectivity might be, or if there's some kind of shared spatial layout. One of the things we've been working on is try to put all this data into a lower dimensional space, and then we can more directly compare some of the activity patterns across different regions, which of course you can't do when they're in voxel space. But I'd love to talk to you more about this idea.
I'm not sure I saw the slides very well, but is it the case you did not get this correlation pattern in V1, which seems to be contrary to what Marina has published earlier?
Yeah, so I found this pretty surprising when I generated these brain maps. A lot of the shared spatial patterns are quite anterior to the occipital pole, which is where a lot of the results have been localized using more univariate methods. I briefly put up some methods where we look at where the time course is synchronized across people, and those were more consistent with where she saw results. So I think it's an open question why the multivoxel pattern that's distributed across space would localize in more anterior areas. I mean, one possibility is that the representations there are more abstract, or they're at some kind of spatial grain that would match what we're testing for. Yeah.
Thanks very much.
Okay, sounds good. No. That one, yeah. Perfect, yeah. Hi. First, I'd like to thank the CHAOSS organizers for giving me this opportunity to present my work to all of y'all. And speaking of CHAOSS organizers, here's Alex Martin. So, if we he does, he looks great. So if we pop Alex's head open and look at his brain, it's fairly easy to find the hand region in his primary motor cortex. And if we have Alex start tapping his left fingers together like this hand region will light up on the right side. However, if we have him keep his hand still, and instead just imagine tapping his fingers together, it's not clear what's going on in this region. If you look through the literature, you'll have several papers that say there's imagining effects in the primary motor cortex, and several others that say there's not. And perhaps we could better answer this question if we were able to look at layer-specific activation in the hand region. And I say this because we know quite a bit about the laminar circuitry in this region. So there's 6 layers in this cortex. For the purpose of this talk, I'm going to simplify that and just say that there are superficial layers and there are deep layers. And we know that the superficial layers get cortical input, so they talk to other cortical regions, and they also have output to the spinal cord to initiate the action. while the deep layer is output only. So this basic circuitry lends itself to an easy prediction. If you have somebody tapping their fingers, then you should get activation in both the superficial and deep layers. You should get this like nice double peak response profile across the layers. And by contrast, if you have someone simply tapping their fingers, you should get just activation in the superficial layer. So this is great, but the problem with measuring these effects across the layers is that this cortex is only about 4 millimeters thick, and so traditional fMRI methods don't give you the spatial resolution to look at this. That's an easy enough problem to solve if you have a 7T scanner, which we do at the NIH. So we have sub-millimeter voxels, and you can see here that we have several voxels spanning the cortex here, so we can look across the layers. However, if we're looking at just the bold response to someone tapping their fingers, we don't get that double peak response. We get lots of activation in the superficial layers and in the sulcus. And that's because bold response is a measure of blood magnetization. And blood magnetization is dominated by these large unspecific veins in the superficial layers of the cortex. So we have to get past this bold problem. So in our study, we used a method called VASO, which stands for vascular space occupancy. And instead of measuring magnetization, VASO measures blood volume, changes in the blood volume. So in this example, you see that this mostly black image is now turned mostly white, because as blood flows into the region, the veins increase in size. And that's basically what VASO is measuring. And you can see that most of that action is happening not in these large unspecific veins, but in these small arterioles that you find in the layer, all across the layers. So these layer-specific veins across the cortex. And now, if we measure the response to tapping with the VASO, we see a different pattern. Now we see nice activation, a nice strip of activation in the superficial layer, and another separate activation in the deep layer. And as you can see now, we've gotten rid of a lot of this bias, and there's a lot less activation in the sulcus as well. And so we can compare these two activations. This is the same data that's just been upsampled by a factor of 4. And the reason we do this is to get a nice smooth response profile across the layers. And so now we can ask if we get the double peak in these regions. And so we use a functional localizer. This is, in fact, the functional localizer of one participant. We draw an ROI, something like this, and then we turn that ROI into 20 equal layers. Now, I know there are only 6, but this gives us a nice, smooth response profile. And for the reasons I just described, the bold signal is almost entirely in the superficial layer, and it tails off as you go to the deep layer. I mean, there's a little bump here, but it's very small in comparison. And by contrast, in the VASO, now we have this nice double peak response profile with activation in the superficial and deep layer. However, before I move on, I want to bring your attention to this specificity sensitivity trade-off between the bold and the VASO. So you've seen the specificity, but if you pay attention to the y-axis here, the percent signal change in the bold is honking. It's huge. And the blood volume, the VASO signal is much smaller. So again, these are data from one participant's functional localizer. We've had 5 participants also perform or complete experimental runs. And in those experimental runs, we had trials where, again, they tapped their fingers together. Other trials where they kept their hands still and imagined tapping their fingers together. And another trial where they wiggled their toes. And we had them wiggle their toes because there should be no activation in the hand region when you wiggle your toes. So it's a good comparison. And finally, rest trials that served as a baseline. Okay, so now across the 5 participants, again, in the bold signal, you get most of the activity in the superficial layer, and it trails off in the deep layer. There's no response to toes, which is good. And now we can ask what's the response to the imagined tapping condition? And we see, as predicted, a slight bump in the superficial layers and no difference in the deep layer between the toes and the imagining. And so we can quantify this. I quantified this by finding the peak response in each of the layers, the superficial and deep layer. And then I further took the response on either side of it and just averaged them together, just to have more information there. You really get no difference if I would have just stuck with the peak. And what you see is a significant response in the bold signal for tapping in both the superficial and deep layers, but no significant response for any of the other conditions. So no effective tapping or imagining tapping when you look at the bold signal. So what about the vaso? So in the vaso, again, you get this double peak. It's slightly attenuated here, but it's still there. Again, no response to toes. And when we plot the response to imagined tapping, again, we get this bump in the superficial layer, but not in the deep layer. And I quantified this the same way. And again, you see a significant response to the superficial, to tapping in the superficial and deep layer. But now, crucially, you see a significantly greater response to the imagined condition in the, compared to the toe condition in the superficial layer, but not in the deep layer. So these data suggest that imagined movements are represented in the superficial layers of primary motor cortex. So in the next phase of this experiment, we want to make sure that this response to imagined tapping in the superficial layer isn't due to inadvertent hand movements when we ask somebody to imagine tapping. You could imagine that you ask somebody to start imagine tapping their fingers, and they kind of squirm around in there. So in the next people we're scanning, we're scanning them with an MRI-safe motion detecting glove. And this glove has 14 sensors on it. For the sake of the presentation, I'm only showing you the four most active sensors. And as you'd expect, you see lots of movement when they're tapping their fingers. But importantly, you see no response to, or no motion when they're doing any of the other conditions. And even more importantly, the imagined tapping condition is equal to the rest condition and the toe condition. And I also averaged the range of motion across each condition. And when I do that, you see this striking difference here. Tons of activation, tons of movement while you're tapping, and none during the other three conditions. So moving forward, we can be confident that any significant responses we get to the imagined condition are due to imagining and not just due to inadvertent hand movements. And with that, I'd like to thank everybody else who's worked really hard on this project. And especially I'd like to thank Renzo Huber. He's a postdoc in Peter Bandantini's lab. He's now at Maastricht. And he's really pioneered these layer fMRI type methods. And a really great resource for learning more about this method is his blog, layerfMRI.com. And I also have a poster downstairs if you want to talk more about the nitty-gritty of the details. And with that, I can take questions. Thanks.
Thanks for keeping your time.
You're welcome.
Hi, that's super cool. So now I know what I'm doing when I'm imagining doing stuff.
Yeah.
So just that last bit about using the motion-sensitive glove, as I understand it, you can get muscular responses that are like a kind of prepotent response prior to making a movement. And so you can measure that, electrophysiologically. And I'm just wondering whether that would be a better method, because you could actually be sending a signal to those muscles, this preparatory signal, which you wouldn't pick up with those gloves.
Oh, that's a good idea. So you're saying you look at premotor? Yeah, that's a good idea.
I mean, evoke muscular response.
Uh-huh.
So I just wanted to add that the implications of this is simply that, you know, in imaging, you get people who say, well, look, you get the same area active, therefore these things share the same neural network or the same neural substrate. And the point of this was to demonstrate, I mean, we all know that perceiving and knowing and imagining are different things, and the substrate has to be different. This was just a demonstration that even within M1, within the same area, you get a very different pattern of activity, so that the claim that you're using the same neural substrate may be incorrect. We used M1 only because it's probably the thickest place in the brain. I don't believe that the real action for motor imagery is M1, and we've argued against that. Something important is probably going on in M1, but you know, we'd want to see premotor, we want to see other places.
That's right.
It remains to be seen whether those areas of the cortex are thick enough to do this, but we're hoping. Mine is, but that's atrophy and what have you.
Thank you, Alex.
So I was just wondering if even if you don't see activity on the glove, if you might still be worried that there's muscle activity nonetheless, which is to say that there's still sort of leakage from the brain down to the hand, and it's, you know, it's sub-threshold, doesn't lead to an overt movement, you don't measure with the glove, and I don't know if you've thought about doing electromyography or something just to...
No, that's a good idea.
Well, and to kind of follow it is like, it very well is probably going to be the case... Oh, sorry. But I think the the expectation is that you will have it.
Yeah, I would say if you're saying that in the case of just in general, you're having these twitches across the conditions, right? So yeah, I know I have not thought of using something other than the glove. No, it's a good idea.
Thank you, Andrew.
Thanks.
Which I probed the question whether conceptual knowledge is represented in a map-like format. So there's increasing evidence that spatially tuned cells in the hippocampal formation, which track positional and directional information, might not only encode mental maps of our physical environment, but provide also a metric to code for abstract non-spatial information. So far, evidence came from studies showing a directional modulation of the human bolt response while humans navigated abstract spaces, such as a feature space in a study by Constantinescu et al., where participants imagined morphing of one bird stimulus into another, corresponding to certain trajectories in a two-dimensional feature space defined along bird stimulus dimensions. So while they imagined this morphing process, they discovered a grid cell-like signal in entorhinal cortex and VMPFC. And similarly, a year before, Tavares and colleagues showed the directional modulation of the hippocampal signal when participants navigated a social space that was defined along a socially relevant dimension during a role-playing game. So these studies indicated basically a directional signal in abstract space. But another critical feature of a map-like representation would be information about distances between locations. And this had so far only been demonstrated in humans for physical space, for example, by the Epstein lab that used fMRI adaptation to show the representation of distances between real-world locations, or by a former postdoc from our lab, Lorena Deuker, who showed that hippocampal pattern similarity across objects that were positioned in a virtual maze reflects the spatial distances between them. And in my projects, I wanted to test whether the hippocampus would encode distances in the conceptual space in a similar manner. So to test this, I trained participants on to learn a very simple concept of two symbol categories. So this is one of the symbols. The symbols could vary along 2 dimensions, circle size and opacity. And the diagonal through the space served as category boundary between the two categories. So participants never saw the spatial layout of this concept. They basically learned it in an exemplar-based manner using feedback. In addition to this concept they acquired, they learned to associate 6 everyday objects with specific symbols, so with specific positions in the space, thereby assigning a certain conceptual distance to the objects. In the free recall task, they navigated the space, however without any spatial instruction. So they were instructed to collect a certain object by editing the feature properties of a random start symbol until this matches the symbol associated with the object to be collected. Now, importantly, we randomized the assignment of those objects to the specific positions, and then critically, before and after this learning phase, we showed only the objects in the scanner in a randomized sequence multiple times, identical before and after learning, to test whether the hippocampus would, as a function of concept learning, map the 2-dimensional distances these objects had in the concept space. I did this using 2 analysis approaches, identical to what was done to show representation of physical distances. So first, using representational similarity analysis, I found that the hippocampal pattern similarity across these objects corresponded to their 2-dimensional distances in the space, with higher pattern similarity for objects that were close in the space. And this pattern could not be equally explained by the 1-dimensional distances the objects would have on either feature dimension alone. So this seems to be really a combined representation. Additionally, I used fMRI adaptation to probe this representation and found that the hippocampal signal scales with the distance between successively presented objects in the way that there was a higher suppression of the signal when 2 objects followed each other that were close in the space. We did not observe the first or the second effect in control regions like the post-central gyrus or LOC, where we wouldn't expect such a combined map, and also not pre-learning. This study left us with 2 questions we wanted to investigate further. So first, what is really the nature of this 2D distance representation? So in our case, the feature space indeed defined a concept. However, as the symbols were so reduced, in this case, conceptual similarity also mapped onto perceptual similarity because they didn't have any other feature dimensions. And so one could now imagine that when participants view the objects in the scanner, they would pattern complete to the associated symbol, and what we actually measure as a 2D signal in the hippocampus as a conceptual map is actually the similarity of the input to the hippocampus. We ruled this already out a bit by showing that there is no reinstatement of this perceptual space in the sensory areas like LOC, where we would predict it, that are involved in processing orientation, size or opacity. However, we wanted to test this more explicitly in a follow-up experiment. Our second question was, well, if this is a conceptual representation, then there should be information about the categorical boundary. And we indeed observed a trend, however, it was quite weak, and we assumed that maybe this is due to the fact that during our very long learning process, categorical information was only task relevant once in the first categorization task, but the other tasks were more important to encode really the specific feature-based information. So I repeated this experiment with two adjustments. So most importantly, the symbols I used now had 3 feature dimensions: opacity, frequency of stripes, frequency of dots. Again, only 2 dimensions were conceptually relevant, and the diagonal in this 2-dimensional concept space again served as category boundary between A and B. The third feature dimension varied, but it didn't contribute at all to the concept. Again, they learned to associate 6 objects with 6 specific positions in this case 3-dimensional feature space. And this basically allowed us to disentangle whether the hippocampal signal would track the 2-dimensional conceptual distances or the 3-dimensional perceptual distances. And I also positioned the points in a way that the 2 predictions are not correlated, so 0.1. So this is the first adjustment. The second one relates to the free recall task or navigation phase. So this time I also wanted to make categorical information task relevant. Therefore, the instruction changed to collect the object of a certain category. So they could choose, but they were instructed to collect every position equally often. So they basically had to combine their specific feature-based knowledge with the categorical knowledge. Again, before and after this learning phase, I showed only the objects in the scanner to probe whether the hippocampus would encode the 2D or the 3D distance. Also, after the whole experiment, I had a control test where participants were asked to reconstruct all 3 dimensions of the 6 symbols. And this was done to make sure that memory between the 3 dimensions is not different, because only 2 were conceptually relevant, and we see there is no memory difference between across the 3 dimensions. Okay, so now coming to our main question. I used again fMRI adaptation. What I did for analysis is in one GLM, I included as a regressor the 2-dimensional distances the objects would have in a concept space. A second regressor, the 3-dimensional distances the objects would have in a feature space, and I took the contrast of that. And what I find is that the hippocampal adaptations is significantly better predicted by the conceptually relevant 2-dimensional distances. So now one could say, yeah, maybe the difference between the 2 predictions is not just the conceptual relevance, maybe it's just complexity. And to rule this out, I conducted 2 control analyses where I basically ran the same analysis, but I derived my 2D predictions from a combination with the irrelevant axis, so either X or Z or Y and Z. And when I do this, the effect disappears. So this shows that indeed the hippocampus seems to map these objects for the purpose of concept learning in specific. I did not find this effect in control regions or in our pre-learning. And also I could replicate it again using the representational similarity analysis as an approach. Well, then coming to the boundary question. So we observed now that also the categorical information was equally task relevant, a representation of the category boundary, in the sense that the hippocampal signal scaled with the distance of each object to the boundary. This effect was not present in control regions or pre-learning. So we think this is quite interesting because information about the boundary only emerges from a combined representation of these 2 feature dimensions into a space. Also, it's coherent or with prior work. So you have to think of then during categorization, uncertainty is always an inherent factor because the closer the objects are to the boundary, the more uncertain they are. And this, what's called decisional uncertainty, was also shown to be processed by the hippocampus before. So to sum up these 2 findings, what our conclusion would be is that the hippocampus encodes a combined space of specifically conceptually relevant feature dimensions. So thank you for your attention. Can take questions now.
Sorry, I'm a bit confused about what it is that is being represented here. So on the one hand, you've got your geometric patterns that you've set up along your 2 dimensions of variation, density or side of the circle. And on the other hand, you've got these objects that you've associated with those. What I'm trying to understand is whether the similarity space you're saying is in hippocampus what I'm trying to understand is whether the similarity space that you're probing in hippocampus is reflecting the associated geometric figures, or whether it's reflecting the combination of the geometric figure and the object.
I see.
Because if it's the combination, then the obvious thing to do, I think, is to manipulate the similarity of those real-world objects and look to see whether that similarity can now also warp the geometric space defined by the dots and circles. So I wasn't sure what you're saying this is a similarity space of, the geometric stuff, the second column in the left, or the objects that are associated with those, or the combination.
Okay. So first of all, the reason why we associated them is that we could randomize the assignment and use them to read out the space in the scanner, such that we do not have any visual similarities that could define our distance effects, or for the RSA, where we subtract basically our pre-learning matrix from the post-learning matrix, we can even rule out any visual or semantic relatedness the single subjects have. So this is why we have this additional step with the associations. That's why we cannot show the symbols themselves in the scanner. And then now I would think, given results of both studies, that what we measure is not a pattern completion due to the associated symbol and something based on that, because then we would also see a reinstatement of this space in the sensory cortex. That's what you usually find, and I tested for that in LOC in places where usually opacity or size are decoded, and I do not find that. So I would assume what we measure indeed is here abstract mapping of the objects into this conceptual context. So also with regard to what other people find with these effects of distances or direction in entorhinal cortex as compared to the hippocampus, I would assume that maybe just the plain structure might be encoded in entorhinal cortex, and what the hippocampus is doing, what it's also known for in other cognitive fields, is that it's therefore the specific binding of the object identity to the structure, to place very specific things into a context. That's what I assume is the signal about.
So just one quick question of clarification. So in this slide here, the navigating the concept space, when you're probing this is a form of, is this like fMRI adaptation kind of duration between the collect A object and then showing that probe picture? Like what's the amount of time?
So this is just a behavioral task.
It's just a behavioral task, okay.
Just a behavioral task. I do not scan this navigation phase. This is just basically a free recall task to improve their learning a bit, because it's more efficient than just
How does it go, the part where you're measuring the magnitude of adaptation in the hippocampus?
What the task is. Okay, so they just see the objects in a randomized sequence and they perform an orthogonal task just to maintain attention. So they basically press 2 buttons for whether there's a target object or not.
Okay, but by adaptation, I'm still trying to understand, by adaptation, are you using that label in sort of fMRI adaptation, or is that?
Yes.
Okay.
Yeah, just,
all right, good.
Just the signal decreases when in repetitions of objects that are close or far in space, but that doesn't relate to the design at all.
Okay.
They're just randomized in
Then I do have a question. So the idea that a lot of people have about what the hippocampus does is that it's not representing lots of different objects in terms of relationships and overlapping code. The predominating idea among a lot of people is that you're sparsifying everything. In other words, you are trying to make a binding between cortical representations that will have no interference across different episodes such that they will not get confused. So I'm wondering how you relate what you're looking at here, where you're essentially proposing something like an overlapping code across related things, to that other proposed function of the hippocampus.
You mean pattern separation? But I
pattern
think that
separation.
both pattern completion and pattern separation exist. So it depends on the relevance of what you're doing then, right?
Do you see what I'm saying, though? If you're representing multiple objects in the hippocampus and you're getting a similarity effect whereby you present one and then very short time after present a related one and show a relatively decreased response, the normal logic of fMRI adaptation is that there's some kind of neural overlap between the first thing and the second thing.
For example, could be if spatial cell types code for this kind of information, then one option is to imagine that the objects are encoded by place cells and they have overlapping place fields explaining the adaptation effect, like in the studies where they find the representation of distances between real-world locations. That's one explanation, but of course I don't know. But yeah.
Okay.
Maybe with the right text. No, yeah, okay, thank you. Thank you.